This second file of 23C starts with the large intestine, but we will refer back to the small intestine quite often. The large intestine has rather taut, it looks a bit like a rope or a tendon, but it actually isn't. It's a collection of three longitudinally oriented smooth muscle bands, and it's called the Tenia coli, or if we were to pluralize it, it would be Teniae. I actually don't care, either one's fine. And because it's relatively taut, it almost acts like a drawstring and ends up creating this kind of scalloped looking pattern in underlying smooth muscle. And each of those pouches, if you will, is called a haustrum, or collectively they are haustra. Another thing that we see associated with the large intestine is hanging primarily off of the Tenia coli are these sacs, these adipose-filled sacs that I think make the large intestine look like it's a melting candle, and are just kind of just disturbing. They're gross. They're called epiploic appendages. And they're actually sort of outcroppings of the visceral peritoneum that's in contact with the large intestine. If we zoom in quite a bit on the large intestine, then we'll see that the mucosa is it's continuing to be simple columnar epithelium until we leave rectum and enter the anal canal, and then we'll transition back into non-keratinized stratified squamous epithelium. Recall that the small intestine features circular folds, villi, and microvilli. Those surface area increasing features are not present in the large intestine, although very few things do get absorbed across the wall of the large intestine. There is no digestion occurring, or at least there are no new enzymes that are being sent into the lumina of the alimentary canal and the large intestine. And then there are even more goblet cells lining the large intestine than we've seen in a lot of the other organs. So the portions or divisions or regions of the large intestine include the cecum, which we talked about in class, kind of a blind corner, very, very reduced with respect to a lot of other animals who absolutely need a cecum, which they use to store partially digested food and prolong digestion, prolong absorption. We have a cecum just vestigially. It's not really doing much for us. It's certainly not acting as a storage bag. And hanging off of the region that we call cecum is the appendix. And within the appendix, we see a lot of mucosa-associated lymphoid tissue. There are all sorts of ideas about what the appendix may do for the body, but because there's a good concentration of malt, excuse me, we can infer that it probably has some immune function. But another possibility is that it kind of provides backstock, that it provides housing for beneficial bacteria so that we can recolonize readily following maybe sickness, especially diarrhea. And the appendix is a bit twisted and skinny, and that's one of the reasons why it's especially vulnerable to being plugged or blocked. Colon is actually just a synonym for the bulk of the large intestine. And you already know that we have an ascending portion, a transverse portion, a descending portion, and a sigmoid portion in that order from proximal to distal. And the ascending colon runs up, obviously, the right side of the body. The transverse colon runs across and is relatively superior. It connects to descending colon, which runs down, obviously, the left side, and then we have to come back to center because we're going to meet the anus, and the anus is right along the midline, obviously. So sigmoid colon is the S-shaped portion that brings us back to center. Where ascending colon meets transverse colon, that turn that has to occur is called the hepatic flexure. It's called the hepatic flexure because it underlies the liver. Whereas where transverse colon meets descending colon, that turn is called the splenic flexure because it underlies the spleen. And then the last portion of the large intestine is the rectum, and inside the rectum are three valve-like walls that allow us to pass gas without pooping, which is something I'm very thankful for, but probably you're not aware that those valves are there in order to get that done. And then the very last thing that we'll talk about is the anus, okay? But I think I mentioned in another lecture that I'm going to have to show you in person how the anal sphincters work. I need you to watch my hands while I explain it. So I'll only touch on it here. Oh, I just said I'd touch an anus. Gross. Hey, slide 3. I can see that embarrassingly reduced cecum. I can see appendix. Okay, which often in models is shown hanging down. Okay, either actually can be the case. Here's the tail end of the ileum. Here's the ileocecal valve, right? Here's our Taenia coli. Here's an epiploic appendage. Here's a haustrum. Here's ascending colon, transverse colon, descending colon, sigmoid colon, rectum, all right? And this corner, hepatic flexure. This corner, splenic flexure. Okay, we can see some mesenteries in the background. Moving on to 4. Right, some things that weren't labeled on 3 are now labeled, but we just went over them. Things like haustrum and Taenia coli and such. Okay, if we move to slide 5, here we can see rectum and anus, and we can see one of the 3 rectal valves, and they're not complete valves in the same sense that, say, the valves of the heart or valves of various vessels are complete closable gate-like valves. These are more like intrusions that slow things down and kind of create obstacles and blockage. And again, these allow us to pass gas without passing feces, so that we can eliminate one convenient thing while not eliminating something that's not so convenient to eliminate just anywhere. We want to be able to choose when and where we pass feces. This portion here is called anal canal, and this opening proper is called anus. But you can see here that both the anal canal and anus proper are flanked by muscle. And this muscle here is actually part of a ring. So imagine the ring is coming out of the screen toward you, and now it's wrapping back into the screen. It's going into the screen away from you and wrapping back. So I was thinking a complete ring. And the external anal sphincter, the internal anal sphincter, the one I was just pointing out, it is made out of smooth muscle. Whereas this block here is also a ring. And even though both rings, both sphincters, are inside the body, this one is referred to as the external anal sphincter, and it is made out of skeletal muscle. And I'll explain how those work together in class. If you move...
Brett, honey, can you turn the light on over me? I'm still recording, by the way. So, since we're not in class, maybe I can show you my hands now. Maybe. It's debatable. Good grief. Okay. So, in class, I allow one of my hands, one of my fists, to represent an internal anal sphincter comprised of smooth muscle. Okay? And the other hand, I wrap around that. And that other hand, therefore, represents the external anal sphincter made out of skeletal muscle. Okay? Well, I have, as you well know from 241, willful control over skeletal muscle. My skeletal muscle is willable. On a bad day, I lose that control, right? But skeletal muscle is willable, okay? Smooth muscle is not, all right? It's gonna relax, contract without your will, okay? And so what happens is when your rectum fills with a stool, because a rectum is really just a kind of a storage tank for a stool, that pressure, that distension, is detected by stretch receptors in the rectum, and that sensory input is sent to the brain, okay? And the brain relays out or sends out two motor plans. And one of those motor plans is sent to that internal anal sphincter, that smooth muscle sphincter, and it says, hey, relax. Okay, now watch closely. I'm going to relax this smooth muscle sphincter. What, you can't tell? How come you can't tell? Because this outer skeletal muscle sphincter is still contracted. It hasn't relaxed, okay? The other motor plan that gets sent out is really to make you aware, hey, as soon as you can, put yourself in a place where it's safe to eliminate a stool. Make sure there are no predators around. And eliminate, shine a light on, eliminate that stool, okay? And it's not until, at least on the good days, that I will this ring, this skeletal sphincter, okay, this external anal sphincter, to relax, that the already relaxed internal anal sphincter can actually widen that lumen to allow the stool to pass. Okay, it was relaxed before that, but this external anal sphincter acted as a clamp. And it's not until we relax that clamp that the internal anal sphincter can also open up. Okay? Sorry for the interruption.
To slide 6. The only thing I'm trying to make you aware of by having this hyper-labeled image is that there are actually a lot of things, a lot of different features that we could point out, but we're only being held responsible for really anus, the sphincters, right, the valves, and certainly rectum. Okay, if you go to slide 7, which is where I am now, we house in our alimentary canal over 1,000 different types, species, varieties, if you want, of bacteria. And the number of bacteria, like the number of bacterial cells or bacterial bodies, if you prefer, that we house inside our bodies outnumber our cells 10 to 1. Well, each of our bodies is made out of trillions of cells. So multiply that by 10. That's a lot of little, well, hopefully buddies. Okay? And when we're healthy, the bacteria that we house in the alimentary canal, they balance each other out. They keep each other in check. Nobody sort of overpopulates or outcrowds anybody else. And those bacteria actually do a lot of beneficial things for us. Not only do they keep each other in check, but they help to keep any disadvantageous or even harmful bacteria in check. So in that sense, they outcompete bad guys. Okay, and any bad guys that do attempt, or even good guys that do attempt to sort of breach the wall of the alimentary canal and enter the circulatory system, well, all along the way we've been mentioning sort of checkpoints that disallow that. Also, the bacteria that we house in our gut, primarily our large intestine, by the way, they can perform digestive processes that we can't, including fermentation, but not limited to. In fact, the cellulose that we ingest, even though we can start digesting it, we can't successfully finish digesting it, whereas there are bacteria that can. So we get some nutrition out of cellulose, not a lot, but we get some nutrition out of cellulose thanks to those bacteria. Bacteria also help us break down other types of carbohydrates. They help us break down mucus, and thanks to the bacteria that we house in the large intestine, we can make righteous farts. So that's a big plus. But arguably, most important is that the bacteria in our large intestine, some of them, of course, are capable of synthesizing vitamins that we either don't get enough of in our diet or can't make ourselves. And that is where the sort of exception, or yet another exception, to the idea that the small intestine does all of our absorption. We established that there are some things, like alcohols, that can be absorbed across the wall of the stomach. Well, the two things that primary things that can be absorbed across the wall of the large intestine are water. In fact, that's one of the chief functions of the large intestine, is to reclaim water, because we've been putting so much water into our foodstuffs along the way, but also to absorb certain vitamins that we can't ourselves make or didn't get in our foodstuffs. Okay, and usually those vitamins are well, they include vitamin K and some of the vitamins in the B complex. Okay, now vitamins that we do get in our foodstuffs, vitamins that we ingest, or vitamins that we can make along the way, those are ingested in the ileum. So we are not ingested, sorry. absorbed. So be careful. Some of the vitamins that we ingest, okay, they're absorbed across the wall of the ileum, and only the ileum. We don't absorb vitamins across the wall of the duodenum or the jejunum, whereas the vitamins that beneficial bacteria make, we absorb across the wall of the large intestine. Those are vitamins that we did not ingest ourselves. Okay, I am on slide 8. What passes to the large intestine will remain there for as many as 24 hours. That's in addition to whatever time foodstuffs spend in our stomach and our small intestine. And so on the low end, your alimentary canal has at least that day's meals still housed within it by the time you ingest the next meal. That's amazing. Now, in the large intestine, recall, we are not doing any digestion. We're not doing any digestion. But our beneficial bacteria that we're housing, they may do some digestion in our large intestine. And then also vitamins that those beneficial bacteria make, where we will absorb across the wall of the small intestine. Any water that I think I just said small intestine, I wanted to say large intestine, but any water that the small intestine didn't reclaim is going to be reclaimed by the large intestine. So the large intestine is going to make sort of the last effort to get as much water back as possible. If we didn't reclaim water, then we would desiccate quite rapidly from all of the water that we put into our foodstuffs. And then we're also going to make an attempt to reabsorb electrolytes, especially very important ones like sodium and chloride. Okay. The other big function of the large intestine is actually a reflection of that water resorption. The more water we resorb from the residue, the stuff that we couldn't digest ourselves, the more solid that residue becomes. And so the other function of the large intestine, the other main function, is stool formation and, of course, moving foodstuffs through. Okay. Well, how does the large intestine move stuff through? In part, it relies on haustral contractions. Most of the contractions that are achieved by the large intestine occur when the haustra contract in sequence, and they do this in response to the stimulus of distension. And even though the haustra are doing this in a very sequential manner, this is considered segmentation. Mostly that occurs in the ascending and transverse colon. Now, when there's food in the stomach, that actually also acts as a stimulus and initiates what's called the gastrocolic reflex. When food arrives in the stomach and the stomach becomes distended, then our large intestine will experience what's called a mass movement, which are slow but very powerful waves of peristalsis that we typically experience 3 or 4 times a day. So you may notice that after a meal, or even sometimes during a meal, that's when you have the urge to pass a stool, and that's why. The descending colon and sigmoid colon they much more so act as a complement to the rectum, and the rectum is a storage tank. I'm on slide 9. We want to start talking about how different types of macromolecules and nutrients are broken down, and we'll spend a lot of time on how they're absorbed. Okay, digestion is catabolic, breaking up rather than building. Right, we already know that in the absorptive state, we expect to build more than break down, but of course we have to digest first before we can build anew. And many of the digestive processes that we pursue start with hydrolysis, adding water to sort of dissolve, if you will, chemical bonds. Absorption, we already know, is the movement of nutrient monomers and small nutrients from the lumen of the alimentary canal into the circulatory system. Tight junctions are numerous along the alimentary canal, and they prevent the entry of pathogens that would otherwise access our circulatory system, but they also ensure that molecules have to pass through mucosa rather than in between the cells comprising mucosa. So pass through epithelial cells rather than squeak in between epithelial cells. Okay, and at some point along here, I can't even remember when, I pointed out the apical surface versus the basolateral surface that we see along the course of the alimentary canal. Apical, lumen; basolateral, closer to the circulatory system, has more direct access to circulatory system. Lipid molecules are absorbed through both the apical and the basolateral surface, though they can pass through without spending any ATP, because our plasma membrane is already phospholipid, whereas other molecules, especially those that are large and or polar, they have to be actively transported. Most nutrients, I don't like that. Oh, no, I do. Yes, I do. Never mind. Sorry. Weird. Most of our nutrients have already been absorbed by the time chyme or foodstuffs reach the ileum. And remember, the ileum is the last portion of the large good grief, small intestine. It's been a long weekend already. I'm looking forward to the break of the week. Okay, so I'm on slide 10, where we'll first talk a little bit about the digestion of carbohydrates, and then when we go to slide 11, we'll talk about the absorption of carbohydrates. So they're kind of coupled together like that. We already hopefully understand that we only can absorb monomers, monosaccharides, and the carbohydrate monomers are, as you hopefully know, glucose, galactose, and fructose, whereas other carbohydrate molecules you may be familiar with, like sucrose, is actually a polymer, at least a disaccharide. Well, in the mouth, we start breaking carbohydrates, complex carbohydrates, down. We break them down into oligosaccharides. Oligo means few, okay, and then saccharide is the fancy pants term that means carbohydrate part, okay. So an oligosaccharide is made out of a few carbohydrate parts. And disaccharides, which means a molecule that's made out of two carbohydrate parts, right? Ultimately, we want monosaccharides, one solitary carbohydrate part. And we start breaking down some of our ingested carbohydrates again in the mouth, primarily with the help of amylase and saliva, okay? And then we hit our carbohydrates again in the duodenum by exposing them to pancreatic juice, which also has amylase. And those two things, amylase in general, but from those two sources, saliva and the pancreas, are going to break down starch and things that we ingest as disaccharides, okay? Whereas we still need to break down oligosaccharides and disaccharides. Again, we want to get monosaccharides. And so the final step that's necessary is achieved via brush border enzymes. Remember, these are enzymes that are secreted by the microvilli of the small intestine's simple columnar epithelium. And so these brush border enzymes help us to break down even oligosaccharides into disaccharides, but we want to finally break these down into monosaccharides, and that's what our brush border enzymes achieve. Okay. Oh, I didn't even realize these three were already on here. Sorry. All right. I'm moving on to 10. In 10, the illustrator gave us at least a couple of, small, but still polymeric carbohydrates. Okay. And we want to break these down. So brush border enzymes are gonna help us to sort of finalize our efforts and get single solitary units that are monomers. And let me see if I can trace one of these for you. There's a monomer. Okay. Well, just because we have finally digested carbohydrates down to their monomer state doesn't mean that we've actually received these in the circulatory system, and so we now need to achieve absorption. And carbohydrate monomers are absorbed via I'm going to write this bigger on the slide because it's so tiny in this caption. A process that's called co-transport, which we'll actually revisit in a subsequent chapter that doesn't address directly digestion. And specifically, the carbohydrate monomers or monosaccharides, they are co-transported with sodium. So yet another function of sodium in the body. They are sort of having to hold hands, if you will, in order to pass through the apical surface of the mucosa in the small intestine. And then here's the basolateral surface. Right? But why would they want to cross the surface in the first place? Well, if we sort of skip ahead and look at and by skip ahead I don't mean that I change slides, but if we skip ahead and look at the basolateral surface, we can see, oh, there's a sodium-potassium pump embedded in the basolateral surface, which is kicking out sodium in exchange for potassium, just like we learned in neurons, for instance. And there's another channel protein that's kicking out monosaccharides, okay? And kicking out monosaccharides means that the circulatory system now has access to them, but it also means, along with kicking out sodium, that we are establishing a concentration gradient where, because we're pumping things out at the basolateral surface, those same things want, yes, I have my quotation fingers up, to enter the cell at the apical surface. They're following their gradient. So that's pretty brilliant. That's how co-transport works. It really just sort of borrows the gradient that sodium potassium pumps are constantly not just generating but maintaining. Very cool. Okay, on to slide 12. Same gist. We'll talk briefly about the digestion of proteins, and then the absorption of those monomers. And we certainly get protein in our diet, but we also will, to some degree, break down the enzymes that participate in digestion, and even just protein products that we encounter along the way. And proteins are first broken down into polypeptides, so not quite as long as, say, the whole protein put together. Large first, then small, and then polypeptides are broken down into peptides, okay? We will get some dipeptides and some tripeptides, but ultimately what we want are monomers, and the monomers are amino acids, okay? We already know that protein digestion is the specialty of the stomach, okay? Achieved primarily with the protease pepsin, but even by the time we enter the small intestine, we could still have some di- and tripeptides. And so, again, brush border enzymes are gonna function in sort of finishing the job. All right. I'm on slide 13, where the illustrator has given us, some small peptide formations, but also, because they've been exposed to brush border enzymes, we're going to see monomers. There's a monomer. All right. And if you look carefully at apical surface and basolateral surface, oh my gosh, this looks exactly the same as the mechanism for carbohydrate absorption, because it is. In other words, in order to absorb amino acids across the mucosa and make them available for the circulatory system, we rely on co-transport with sodium. And the gradient that we need in order to sort of attract sodium into this columnar cell, or these columnar cells, is achieved via a sodium-potassium pump. So exactly the same mechanism, okay? Which is kind of nice. I'm moving to slide 14. Now, fat digestion and absorption are both a bit different than what we saw with carbohydrates and proteins. With fats, with lipids, we tend to see them forming globules in our foodstuffs. Okay, and we expose these to lipases along the way, but it's not really until fat is exposed to bile salts, so in the duodenum. That we start to really see significant emulsification, which isn't exactly, at least it doesn't look so much like breaking up a molecule. It's more like subdividing a droplet, okay? So we're getting smaller droplets, which are actually called fat droplets, okay? Now these droplets are small enough, in comparison to a globule, for various lipases from the pancreas to have access. And those lipases will yield monoglycerides and fatty acids, all right? And then those fatty acids and monoglycerides will kind of couple with bile salts to form what are called micelles, okay? And it's in this form as a micelle that absorption can begin. Okay, the brush border, the microvilli of our beautiful mucosa here, they will claim from micelles those fatty acids and monoglycerides, okay, and transport them through. Primarily, it's just passive transport, but primarily it's they're just following a gradient. Excuse me. And then within the epithelial cell, these monomers will form chylomicrons. And every time I see or say this word, I think of midi-chlorians, which is a Star Wars thing. But chylomicron is sort of a recombination. We're taking these monomers and repackaging them. We're repackaging them with other fatty substances as well as with proteins, okay? Sorry. And it's as chylomicrons that fats are transported from epithelium all the way into lacteals. Okay, we're not going to send our lipids into the circulatory system, at least not yet. Slide 15 is sort of backwards. With the other nutrients, we've been talking about digestion first and then absorption. But this slide is a little more concerned with digestion at its second. So that's backwards to some degree. But yeah, we do expose fats to lipases in saliva. We do expose fats to lipases in the stomach. But it's not until the duodenum that fats really get hit. Okay, on to 16, the digestion of nucleic acids. So even though I agree that DNA is a small part of any cell, we ingest enough cells that we could claim nutrients from DNA and RNA, and so we do. You know, might as well bother. So It's nucleases in pancreatic juice that focus on nucleic acids and free up nucleotide monomers. Okay, and in the small intestine, in addition to being exposed to pancreatic juice, those those nucleotide monomers are exposed to brush border enzymes, and brush border enzymes will sort of finish the job by dividing nucleotides into nitrogenous bases, sugars, the sort of rail, if you will, of the DNA ladder, and phosphate ions. All right. And then those products are actively transported across the epithelium in special sort of nitrogenous base carriers or transporters. All right. So primarily, nucleic acids are going to be digested in the duodenum. I'm on slide 17. But this is actually review, but it's important. We absorb vitamins across the small intestine and across the large intestine. The difference is, did we ingest those vitamins, or are beneficial bacteria that we are housing making those vitamins? Okay, if we ingested them, then we're going to absorb them across the wall of the small intestine, specifically the ileum. If bacteria produce them, then we are going to absorb them across the wall of the large intestine. And those that are generated by bacteria tend to be, again, vitamins K and parts of the B complex. Those that we ingest or sort of make along the way include, and therefore absorb via the ileum, include some B vitamins, C, D, E, and of course some K. So that means that the beneficial bacteria in our large intestine are simply supplementing what we ingest, and needfully so. Some of the vitamins are fat soluble, some of them are water soluble, and therefore they will enter or cross, I should say, the epithelium of the small intestine via different means. But certainly we should know that vitamin B12 has to couple with intrinsic factor in order to be endocytosed by simple columnar epithelial cells of the small intestine. Slide 18. We need to reclaim ions from our foodstuffs, and then we're also claiming ions that we've ingested from our foodstuffs. And those are, for the most part, actively transported along the whole length of the small intestine with, keep in mind, it all depends on the ion. So iron and calcium, they tend to get absorbed early on across the wall of the duodenum. Sodium, as you know, is going to be co-transported with carbohydrate monomers and protein monomers. So that's going to happen mostly in the jejunum. Chloride is transported actively, but there's no primary site of that activation, or that transport, I should say. Potassium simply follows its gradient, and that is a reflection of whether or not we're absorbing a good deal of water. And then calcium absorption in terms of like how much we achieve, we already know is a reflection of vitamin D availability, as well as the influence of parathyroid hormone. I'm on slide 19. So as we move foodstuffs through the alimentary canal, we actually add 9 liters of water. That's crazy. 9 liters of water. And that's per day. We actually have water flowing in both directions across the walls of primarily our small intestine and large intestine. But it's the movement of solutes that creates a net movement of water from lumen to circulatory system. And I think you've already mastered that. You know that where sodium goes, water goes. So that's key. And that actually is the explanation for some diarrhea. Diarrhea can occur for a variety of reasons, but often whether the mucosa is damaged or the chemistry is wrong or whatever, often what's happening is the osmotic balance in the lumen of the alimentary canal is off, and the water follows the solutes, and hence you get a watery stool, which is the very definition of diarrhea. We reabsorb about 95% of that 9 liters across the wall of the small intestine, and then the large intestine does the rest. And then we also ingest about 1.5 liters every day. And then in a healthy stool, we would maybe find about 200 milliliters. So we do a really good job of not just reclaiming what we put in, but also what we put in along the way, but also reclaiming. Good grief! I can't talk right now. Also claiming what we ingested. Okay, so we've really taken a lot of water. Anywho, we're good.